Inside a child's bone marrow, a leukemia cell is trying to copy itself. It needs raw material, especially the molecular building blocks used to assemble DNA and RNA. Given a molecule that looks close enough to the real material, and the cell may pull that imposter into its own machinery. Then the copy process begins to fail from inside. That strategy helped produce one of the first medicines to create meaningful remissions in childhood leukemia. It did not arrive from guessing at random bottles. It grew from a question: could a drug be designed around a biochemical difference between a diseased cell and the person carrying it? One of the people who pursued that question was Gertrude Elion. She would help create medicines for leukemia, organ transplantation, gout, malaria, bacterial infection, and herpes viruses. Her name eventually appeared on dozens of patents. Yet when she began, laboratories repeatedly told her that a woman would distract the men. Her story is not a lone genius fairy tale. George Hitchings built the research program. Elvira Falco, Peter Russell, chemists, biologists, physicians, and patients turned molecules into medicine. Elion's gift was learning to move between all those worlds without treating any disciplinary wall as permanent. Elion was 15 when her grandfather died of cancer. She later described watching the disease take him and deciding she wanted to understand why. She studied chemistry at Hunter College during the Depression, when her family could not afford an expensive university. Graduate schools offered little support, and research employers routinely rejected women. She taught, worked in a food laboratory testing acidity, and pursued a master's degree at night. None of it looked like the beginning of a Nobel career. It looked like someone staying close to science by whatever door remained unlocked. During the Second World War, laboratories short of male chemists finally hired more women. In 1944, Elion joined George Hitchings at Burroughs Wellcome. She also tried to continue a doctorate at night until the university required full-time attendance. Leaving the laboratory would mean abandoning the work that had become her real education. She chose the work and never completed a PhD. In Hitchings' small group, titles mattered less than useful questions. Elion moved from organic chemistry into microbiology, biochemistry, pharmacology, immunology, and virology. The missing degree stopped defining what she was allowed to learn. Hitchings believed cells could be attacked through their metabolism. Purines are essential components of DNA and RNA, but they also move through intricate networks of enzymes and intermediate molecules. Different organisms and diseased cells do not always handle those pathways in exactly the same way. The group synthesized altered purines and watched what happened. A useful analog needed to resemble a natural molecule closely enough to enter the pathway, but somewhere in the process, its difference had to matter. It might block an enzyme, create a useless product, or stop a chain of reactions. This was not modern computer-aided drug design. The pathways were incomplete, and many enzymes had not been characterized. Each compound became a probe. If bacteria stopped growing, or tumor cells changed, the result revealed something about the machinery as well as the drug. Elion treated those results like a mystery. Chemistry suggested a structure, biology showed an effect. A new structure tested the explanation. The team moved back and forth until the pattern sharpened. Instead of asking only whether a chemical killed cells, they asked why one cell was vulnerable and another survived. One analog was 6-mercaptopurine, usually shortened to 6-MP. It interferes with purine metabolism and the production of nucleic acids. Rapidly dividing leukemia cells are especially dependent on that machinery, though healthy tissues can be harmed too. The drug required careful dosing and clinical partnership. In the early 1950s, children with acute leukemia usually faced a brutally short future. 6-MP produced remissions and became a foundation of combination treatment. It was not a universal cure. Resistance emerged, toxicity mattered, and other drugs were needed. A time that had seemed impossible became medically real. Elion followed what happened beyond the laboratory. Families wrote to her about children who lived longer, transplant recipients who returned home, and patients whose pain or infections came under control. She kept many of those letters. That connection changed the emotional scale of drug development. A diagram of a purine pathway could end in another birthday. A slightly altered atom could become a school year a child was not expected to see. The molecules were precise. Their consequences were not abstract. A relative of 6MP became azathioprine, an immune-suppressing drug. An organ transplant creates a biological contradiction. The new organ can save the patient, but the immune system recognizes it as foreign and attacks. Suppress that response too little, and the graft fails. Suppress it too much, and infection becomes dangerous. Azathioprine, combined with other advances, helped make long-term transplantation more practical. It did not create the field alone. Surgeons, immunologists, tissue matching, steroids, and later medicines all mattered. Elion's chemistry supplied one crucial lever. The same research culture produced allopurinol, which reduces uric acid production, and transformed gout treatment, especially for cancer patients endangered by rapid cell breakdown. Other compounds targeted malaria parasites or bacterial folate metabolism. The pattern was broader than any one disease. Find a biochemical dependency. Compare the pathogen, tumor, or immune cell with healthy tissue. Design a molecule that exploits the difference. Absolute selectivity was rarely possible, but relative selectivity could turn poison into therapy. Viruses posed a harder problem because they reproduce inside human cells. Kill the machinery the virus uses, and you may kill the host cell too. Many researchers doubted that a safe antiviral drug could be sufficiently selective. Elion's team pursued acyclovir against herpes viruses. The molecule remains relatively inactive until a viral enzyme, thymidine kinase, adds the first phosphate inside an infected cell. Human enzymes complete the activation. The infected cell helps switch on the drug aimed at the virus. Activated acyclovir resembles a DNA building block. Viral DNA polymerase takes it up, but the molecule lacks the structure needed to continue the chain normally. Viral DNA extension stops because activation happens far more efficiently in infected cells, and the compound favors viral polymerase. Treatment can suppress the virus with useful selectivity. Acyclovir does not remove latent herpes virus from the body. It limits replication and disease. The achievement was not magic targeting. It was layered selectivity built from the viral enzyme, the drug's activation, and the polymerase that accepts it. In 1988, Gertrude Elion shared the Nobel Prize in Physiology or Medicine with George Hitchings and James Black for principles of drug treatment. The scientist who had been denied laboratory jobs and had never completed a doctorate was honored for changing how medicines could be conceived. She always emphasized collaboration. Her career stretched across chemistry, biology, medicine, and the experiences of patients. Rational design did not eliminate experiment. It made each experiment ask a better question. The medicines associated with Elion reached leukemia wards, transplant units, infectious disease clinics, and ordinary medicine cabinets. Their common thread is a disciplined kind of empathy. Understand the machinery closely enough to interrupt disease without forgetting the person around it. If you want to follow another overlooked molecule from a chalk sketch to the lives it changed, subscribe here on the board. Then tell me in the comments which medicine, invention, or impossible experiment we should trace next.